Hello friends, welcome to the student world. Today we will talk about a rarest blood group found first time in India, that is EMM negative. According to Times of India dated July 15, 2022, a rarest blood group is found in a 65-year-old person from Rajkot in Gujarat. He is the first Indian and world's 10th person with this rarest blood group. Now we took a look on a story behind this discovery. According to Sanmukh Joshi, doctor at Samarpan Blood Donation Center, Surat, reported that a person has suffered from a heart attack in 2021. Doctors recommend him blood surgery, so he required blood for this surgery. His known blood group was AB positive, but the surprise is that his blood group didn't match with any blood in the blood center or with the blood of his relative. The doctors postponed the surgery and they referred the patients to Ahmedabad. The Pratna laboratory in Ahmedabad also examined the blood group but found the same result, that his blood didn't match with any blood in their blood bank. After examination, they sent the blood to a laboratory in New York for test. In New York, the researchers tested the blood sample and analyzed it for 1 year, and finally they come to the conclusion that the blood group was rarest blood group with EMM negative phenotype. The ISBT named this blood group as EMM negative. Let's talk about how we determine the blood type of a person. Our red blood cells have proteins and sugar molecules on the surface of RBC. These molecules act as antigens. Antigens are the foreign substances that can evoke our immune system and generate an immune response against that molecule. The presence or absence of these antigens determine the blood group of an individual. For example, in the ABO blood system, the presence of A antigen on the surface of RBC give the blood group A, and the presence of antigen B on the surface of RBC give blood group B. The antibodies are present in the blood against the antigen which is absent on the surface of RBC. For example, if antigen A is absent on the surface of RBC, then anti-A antibodies are present in the blood of that individual. Let's talk about blood group antigens. There are 375 types of blood group antigens which are recognized on the surface of RBC cells. These antigens are classified either into blood group systems or in collections or series. Most of these antigens are classified into blood group systems such as ABO blood group system, RH blood group system, Duffy, etc. The remaining antigens are classified either as collections or series. Series are of two types: high incidence antigens of 901 series and low incidence antigens of 700 series. 901 series antigens occur at high frequency, means these antigens are present in more than 90% of the population. 700 series antigens are of low frequency antigens which are present in less than 1% of the population. Now come to the topic, what is EMM? EMM is a high incidence antigen. High incidence antigen means that this antigen is present in almost all human red blood cells. That is the antigen that occur in greater than 90% of the human population. This RBC antigen is assigned the number 901008 in ISBT 901 series of high incidence antigens. The person which lack this antigen on its RBC has been assigned the blood group as negative by ISBT. Persons having this blood group have naturally occurring anti-EMM antibodies in their blood. Let's talk about molecular basis of this negative blood group. This phenotype negative is due to a mutation in PIGG gene. This gene is involved in GPI modification. GPI is a glycolipid that helps in anchor of protein to the cell surface. antigen is carried to the cell surface by this GPI protein. antigen is carried to the cell surface by free GPI, and some component of this glycoprotein also form some epitopes of antigen. Mutation in this gene is responsible for lack of this antigen on the surface of RBC and result in negative blood phenotype. Discuss about the importance of this blood typing. A fully specified blood type should describe the complete set of antigens that are present on RBC of a person. Before blood transfusion, these antigens should be determined for both donor and the recipient. If all the antigens of donor and recipient RBC match, then only blood transfusion is done. To check whether the antigens are similar or not, the blood of recipient is mixed with the blood from the donor and examined for the agglutination reaction. In agglutination reaction, the clumping of RBC is observed. If there is no agglutination, then the blood group is compatible. If blood type is mismatched, then serious complications can develop, such as acute hemolytic transfusion reactions. In these reactions, in hemolytic transfusion reactions, the RBCs are destroyed by person's own immune system. Thank you. If you like the video, please subscribe.